अभी आपके से पे नाम अब चीज तो बहुत बाहर रखने के लिए
Why rest today? On 7th day exam. Exam is going on. So many of them from 7th day. Okay, so Bolo, saying something. We had already finished with the chapter on cell, structure of chromosome and cell division, no? Structure of chromosome, cell division, we are already done. We were doing some questions the other day and I had asked you some questions to complete. Did you all do that? Any doubt in that? I'll give you 5 minutes. Go through those questions. Take out your books. Go through that question. Last time I asked you to solve and complete those questions. Go through those questions. I had asked you to take in the textbook and solve those questions. All done? Any doubt in those questions? Go through. I'll give you 5 more minutes. Go through any doubt, any confusion in any of the questions. What was the last topic? What other topics are over in your school? Photosynthesis, translation and respiration. Absorption by roots. Okay. And all of you are from the same school? Which school is this you are saying? Hodar. Acha. Photosynthesis, transpiration and absorption. These 3 chapters in a book. What about the rest? Absorption by roots. That is going on. Before the holidays, that's over. Achha. Absorption by roots. Okay, good. Same, no? This school is same. Okay. So in that case, if that is a common chapter, then we'll start with that only. Absorption by roots. No, that's a chapter that you need now. So we are already finished with that chapter on cell division, structure chromosome, that is over. So today in that case we'll start with absorption by roots. Before that, I said I'll give you just 5 minutes. Go through this cell cycle, cell division, structure of chromosomes. 1 or 2 questions I will ask you right now that you have to write in your notebook. Once you are convinced that more or less you are okay with this chapter, then we'll move on. Just 10 minutes more I can devote for this chapter. Then let's start with the next chapter. Diagrams. Are you thorough with the diagrams? Will you be able to draw the diagram, cell division? Okay, now I'll just give you 10 questions. Name the following. If you are done with these questions, if there's anything you ask, otherwise, take out your notebook. Take out your notebook. Just 10 questions to write. The first is, these are all one questions that I'm giving you now. And in between, I already told you, off and on, anytime, any day. जब भी मैं बहुत खुश रहती हूँ मुझे टेस्ट करना बहुत अच्छा लगता है. The day I'm very happy, anytime I will ask for a test. वो तुम्हारा बैच भी हो सकता है, कोई भी बैच हो सकता है. So be prepared with the chapter that is already done here. And you are already aware that the tests of UT are starting soon, no? So you have to be prepared. You are given the test schedule, no? You know? What is the first chapter you are going to be tested on? This chapter. So get prepared. Diagrams, whatever I told you are important, keep yourself fully prepared. First question right now. You will not refer to the text, you will try to recollect and write up. Number one, name the following. So the sub questions you keep writing. All of them are name the following. Answer in one word. Number one, the packing unit of DNA. The packing unit of DNA. You may write down the question so that you remember what answers you are writing. Packing unit of DNA. Number two, the stage of cell division when chromosome duplicates. Stage of cell division when chromosome. You may write down the questions. After that, I'll give you 5 minutes to answer. Stage of cell division when chromosome duplicates. Keep one line gap for the answer. Number three, the longest phase of interphase. Longest phase of interphase. Number four, the process of nuclear division. Process of nuclear division. The stage of cell division. Stage of cell division. Stage of cell division when spindle formation takes place. Spindle formation takes place. The number of histone subunits. Number of histone subunits that form a nucleosome. That form a nucleosome. That form a nucleosome. 5. Repeating unit of DNA molecule. 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 Next, type of cell division that takes place in reproductive cells. Type of cell division that takes place in reproductive cells. Type of cell division that takes place in reproductive cells. The phase of interface where formation of new DNA molecule take place. Formation of new DNA molecule take place. Stage of mitosis in which duplication of centrosome takes place. Duplication of centrosome takes place. Duplication of centrosome takes place. I'll give it just two more. यहाँ पे I think this two or three concepts I'm going to discuss. Number 11, I think that is no. Number 11. Number 11. The type of cytokinesis. Type of cytokinesis shown by plant cells. Type of cytokinesis shown by plant cells. Type of cytokinesis shown by plant cells. And the last one. Type of cytokinesis shown by plant cells. And the last one is stage of cell division in which chromosomes uncoil. Stage of cell division in which chromosomes uncoil. Uncoil. 5 minutes finished. Write one word only. There's nothing to describe. Name the following type of a question they are. Check how many of them you are able to answer. Don't take too long. You're done? Over? Over? I gave you 12 questions. All done? Very good. अगर 6 भी आता है, 7 of which are able to answer, it's very good. 6, 7 भी अगर आ रहा है, that's very good. But I'm sure most of you should be able to, must have written all answers there. ठीक है? So that's not a problem. अभी हमने जब चैप्टर कंप्लीट किया should be able to just एक दो बार पढ़ोगे so you'll be able to remember. Should we discuss now? Done? हो गया ना? We'll start from here. बोलो. First one answer. Read. फटाफट read करो. Read and tell me the answer. हाँ.
The packing unit of DNA.
हाँ.
Cellulose.
नहीं. Packing unit of DNA. What is the name? One word. No. Packing unit of DNA. Nucleosome. Nucleosome. Correct? If you have written wrong. Packing unit of DNA is nucleosome. Spelling याद है? Nucleosome. Nucleosome. चलो next. Chromosomes duplicate. Prophase. Duplication of chromosome takes place in prophase. Third. G1 phase. Longest phase of interphase, longest phase of cell cycle, G1 phase. Fourth, क्या है क्वेश्चन बोलो?
The process of nuclear division.
अरे speak loud, no, you are a boy, no?
The process of nuclear division.
Process of nuclear division. क्या है आंसर? Karyokinesis. स्पेलिंग याद है? Karyo, K-A-R-Y-O, K-I-N-E-S-I-S. The last one is fifth.
The stage of cell division and spindle formation takes place.
Spindle formation takes place. कौन सा?
Prophase.
Prophase. Very good. Prophase. Prophase. Six.
The number of histone subunits that form a nucleus.
Eight. Eight. Very good. Eight. Eight. हाँ? हाँ?
Next question.
हाँ बोलो. Seven.
Repeating unit of.
अच्छा, what are you scared about? मुझसे तो कोई डरता होगा. I don't believe ऐसा भी कोई प्राणी इस universe में है. Why don't you are not able to speak properly? क्यों? आराम से be very free in the class. You should be able to interact with the teacher. That's the correct way how you learn much better. See, मेरा tone ऐसा ही है. Right from birth, perhaps I'm this way only. That's the way I talk. My mother kept on telling me, "यार बेटा धीरे से बोलो, sweet बोलो." I could never do that. But don't go by my words. I am constantly. I'm going to give you this kind of tests and constantly I'm going to brush you this way. There's nothing. You should be free in the class. Doubt है, confusion है, you should speak up. बोलो. Number 7.
Repeating unit of DNA molecule.
हाँ.
1, 3, 1.
अरे कौन सा स्कूल से पढ़ के आए तुम आंतरिक? Repeating unit of DNA molecule को क्या बोलेंगे? Nucleotide. कौन सा कहाँ से मिला तुमको कौन? Repeating unit of the DNA molecule का नाम क्या है? Repeating unit of DNA molecule को nucleotide बोलते हैं ना? So that's what you have to write down. Nucleotide. Next.
Type of cell division that takes place in reproductive cells. Meiosis.
Meiosis का spelling क्या है? M-E-I-O-S-I-S. लिखा? Next. Number 8. Number 8 is number 9 now. 9. हाँ. हाँ. Phase of interphase, phase of cell cycle where synthesis of DNA takes place. Which phase? What are the phases? G1 है. G1 के बाद वाला phase कौन सा है? S phase. S phase में क्या सीखा आपने? S phase में what is the most important event? Synthesis of new DNA. S phase. Synthetic phase. S phase. ठीक है? Next.
Stage of mitosis and its duplication of centrosome.
हाँ. Prophase. Duplication of centrosome, duplication of chromosome, all in prophase. Next.
Type of cytogenesis shown by plants.
Type of cytogenesis shown by plants. There's a word that you have to remember, no? Remember we had talked about that. If it is, now this is a plant, animal cell that I'm trying to show you the diagram again. This is the animal cell. Now if the cell division, if the cytoplasmic division starts from the margin to the center, यहाँ पे margin से center की तरफ आता है ना? This is what is called centripetal. Centripetal. So now this is centripetal cytogenesis. But the plant cell में ऐसे होता है? No. If it is a plant cell, then I have to show you that the cell plate formation starts from here, center, and moving this way, and moving this. That is the movement is from center to the periphery. No, center to the periphery. This is called centrifugal. 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 I hope I'm correct. Just check. Centrifugal and centrifugal. Type of, nature of plant mitosis. No, what I said? Plant mitosis is centrifugal. Correct. Plant mitosis process is centrifugal. Correct. Centrifugal always means center to periphery, center to edge. Plant cell में हमेशा this word centrifugal. Animal cell में periphery to center, centripetal. These are the last one. That's the last.
Yes.
Done.
There is the
एक और बोलो, last बोलो.
stage of cell division in which chromosomes are coiled.
Stage of cell division in which chromosomes uncoil. Coil stage से भी thread हो जाता है. कौन सा? Telophase. Telophase. Name the following is a prominent set of questions that actually in your paper all questions are 1 mark. So these type of concepts you should be very thorough with. In your book, every chapter, especially the definition, अगर तुमको definition नहीं आता है, if you don't know these terms, variety of questions you will not be able to answer. क्योंकि this is the basis on which differences you are writing. This is the basis on which you are identifying that most of your paper will have diagram-based questions, definition, identification, name the following. These are prominent type of questions in your paper. So you have to know all these terms very clearly, very nicely, and for that you have to keep revising. Revise करते रहना है chapters. Now I think we did not discuss this because I just now saw the last part of the chapter. I don't think I have discussed few differences, few terms you are supposed to understand. Let me just complete with this. Abhi, meiosis that you already know, that we already discussed that meiosis is a type of cell division that takes place when the germ cells, reproductive cells. So which are the type of germ cells I am talking about? If I have to talk about the animals, if I have to talk about the animals, I am talking about the cells of the testes, please remember. And I am talking about the cells of the ovary, the male reproductive gland, the female reproductive gland. Is it that the process of meiosis is only happening in the animal cells? No, it's also happening in the plant cells. So if it is in the plant cells, then if it is in the plant cells, then it's in the reproductive organ of the plant, the flower. The reproductive organ of the plant is in the flower, no? Now, in case of the flower, if I have to talk about the male reproductive organ, then I will talk about the anther, the stamen, the stamen. And then here I talk about the ovary, the anther. We have got the stamen, which has got the anther and the filaments, and then we have got the ovary. So here, these are the terms that you have to remember. Reproductive, the process of meiosis. In which type of cells meiosis takes place in the plant, in the animal. अभी एक और concept which I did not tell you is a concept of haploid and diploid. Now let me just talk about the cells of the testis. Okay, I now talk about the cell of the testis, and this cell of the testis has got 46 chromosomes. This cell of the testis, like any other cell of our body, now that can be the cell of the liver, that can be the cell of the stomach, that can be the bone cell, that can be the cell of the skin, anything. Now this cell has got 46 chromosomes, and this cell, when it divides, if it is by mitosis, what do we know? The number of chromosomes remains the same, no? If it is meiosis, I will show you some of this so that you remember the basic differences, but stage drawing is not there for meiosis, not to worry. I am saying that if this is a cell which is undergoing the process of meiosis, then what? Initially we get two cells. Initially we get two cells. And in these two cells, yes, the number of chromosomes remains the same, but this is not the end of the division, where the process of meiosis again happens in another phase. Meiosis is finished in two phases. Then we come across 1, 2, 3 and 4 cells where the number of chromosomes just becomes half. Where we talk about the number of chromosomes would just become this way. Okay. Now, this is a cell which I will say this is a diploid cell. Why? Because it has got full set of chromosomes. Half set and half set, meaning 23 chromosome in pairs it has got. What is the meaning of the word diploid? A cell which has got the full chromosome complement. A cell which has got two half sets of the chromosome. Two half set matlab एक 23 plus एक और 23, no? And the chromosomes are all like this, paired, no? Chromosomes are all like this, paired. So whenever we are talking about the concept of diploid, what is the meaning of that? Define diploid condition. What to write? What is diploid? Diploid condition of a cell means the cell has full chromosome complement. Now, in case of human, it is 46. In case of some other animal, it can be 38. In some other animal, it may be 16. In some other animal, it may be 8. In some animal, it may be 2. So ऐसे भी हो सकता है. Now, whatever is the chromosome number, if that cell undergoes meiosis, the chromosome number will be just reduced to half. 8 to 16. If it is 38 to 19. If it is 20 to 10. If it is 40 to 20, जो भी number है, it will get just divided into, it will just get reduced to half. Meiosis है. I'm talking about meiosis. Meiosis है तो how many daughter cells? That you know already. 4 daughter cells होगा. Every daughter cell will have just the half of the chromosome number of the parent cell. This cell, I will say, this is a haploid cell. This is a haploid cell. What is the meaning of haploid? A cell which has got only one half cell of chromosome. A cell which has got only one half cell. Meaning, इसमें सिर्फ 23 है ना? The other cell is not there. Meaning, if I talk about this cell, it has got just one chromosome from every pair. But इसमें no, the chromosomes are paired like this. सारे paired है. Am I clear? Diploid and haploid. What is the meaning? Diploid full chromosome complement या 2 half cells. Haploid 1 half cell of the chromosome. These haploid cells are majorly the germ cells. What is the alternative word? Gametes. If it is the male reproductive organ, gametes are sperms. If it is the female reproductive organ, gametes are ova. Yeah, ovum. Ovum. Let me write it simply. Ovum. Cells of testes has got 46. Sperms will have 23. Cells of ovary has got 46. Ovum will have 23. So that's the rule. Why? Because these are the cells which will undergo the process of meiosis, where from one cell 4 cells will be developed. Every cell will have just half the chromosome number. Now the question is, are these cells able to divide on their own? No. Why not? Because these cells have got just one half set of chromosome. The other counterpart is not there. This has got just 23. This has got just 23. Can they multiply on their own? No, they cannot. But if I talk about the cell of the skin, every cell has got 46 chromosomes. Every cell immediately can undergo the process of division. These are exceptional cells of our body. These are exceptional cells. When I talk about the plants, they cannot divide on their own. What is the reason? Because they have got just one half set of chromosome. So what are we waiting for? They have to undergo a process known as fertilization. Then they will again get converted into a diploid cell by the process of fertilization. And what will be this single cell termed as? This single cell will be termed as zygote. Is this the concept true for animal and plant both? Yes. Clear, no? These are basic concepts. We don't need to understand the process. I'm not explaining you any process here. Process तुम्हें नहीं चाहिए, but this concept we have to understand. What is the diploid cell? What is the haploid cell? What is the haploid chromosome number in human? 23. What is the chromosome number in the cells of ovary? 23. Cells of ovary में chromosome number क्या है? 46. How many pairs? How many pairs? 23. 23 pairs. Cells of ovary में 23 pairs. No confusion. ठीक है? Any doubt? Anything to ask? Clear है? Plants में ये है. Cells of the anther, cells of the ovary. Same way, this will have 46. I'm sorry. This will have. If I talk about a particular plant, say suppose it has got 18 chromosomes. For an example. Then these are producing the pollen. Then how many chromosomes should the pollen have? 9 each, 9 each, 9 each. Whatever the number, just half. Clear, no? So this is the concept. So एक तो ये है. Now we learn something more, 2-3 terms yet you should try to understand 2-3 more terms. Now एक है, when we had talked about the concept of mitosis, this was not there. Meiosis, generally a type of division which gets completed in 2 phases. Meiosis, a type of a division which gets completed in 2 phases. Now this is the parent cell, and this will first of all undergo the first phase known as meiosis 1, and then we talk about as a result of meiosis 2 we get the daughter cells. No? So did we study something like this when we were talking about mitosis? No. For mitosis there was no phase 1 and phase 2. All the stages were just happening once. When I say meiosis 1, what do I understand? Meiosis 1 में prophase 1 and then metaphase 1, no? And then anaphase 1 and then what? Telophase 1. So this is the first phase. And same like that, phase 2 will also have the second set of stages. Prophase 2, metaphase 2, anaphase 2, telophase 2, and then the phase is over. So this is one difference that you remember. Mitosis and meiosis differences you are asked in the paper. Huh? What are the major differences? Number one, this is the first difference I'm talking about. What is the first difference I say? If it is mitosis, the process of cell division is shorter, has only 4 basic stages. If it is meiosis, the process of cell division is much longer, which has got 2 basic phases, and each phases has all these 4 substages happen. Meaning, prophase happening twice, metaphase happening twice, not that way. So that's one difference. What is the second difference? Mitosis happening in somatic cells, body cells, skin cells, bone cells, liver cells, pancreas. Meiosis happening in the reproductive cells only. So we have been talking about these examples. What is the third difference? Let us try to understand. In the third difference, when we talk about a cell which is going to show the process of meiosis, the chromosomes, they always try coming together. Remember, when I was explaining you a cell which was undergoing the process of mitosis, if I have to randomly draw a cell undergoing the process of mitosis, I will just draw that the chromosomes are this way. We just draw the chromosome just that way, you know. Just randomly you can draw the chromosomes. You need not show them paired up. But if I have to talk about the concept of meiosis always involves this particular process because there are additional proteins present which looks for the maternal and the paternal chromosome. What is the meaning of maternal paternal? Now that the zygote is formed, 23 chromosome coming from the father, 23 chromosome coming from the mother. Let me talk about ये जो यहाँ से एक पेयर यहाँ से एक क्रोमोसोम और यहाँ से एक और क्रोमोसोम, they are forming a pair. One chromosome from the father, one chromosome from the mother. Something same thing I'm drawing here. One chromosome from the mother, one chromosome from the father. What is this pair of chromosome known as? This pair of chromosome, which is a typical feature of meiosis, ये mitosis में नहीं होता. This coming together of the chromosome and then starting the process of cell division, this pair of chromosome, what is this called? This is what is called the homologous chromosome. This is the term that you should remember. What is homologous chromosome? A pair of chromosome which has got one maternal and one paternal chromosome. A pair of chromosome जहाँ पे एक maternal chromosome है, paternal chromosome है, but then the question is that how do they come together? They come together because of additional proteins which get released in the nucleoplasm, and तब जाके उनका pairing होता है. And you have to also understand here one more word. What is that word that you have to also understand? Homologous chromosome undergo a process known as synapses. I think synapses that is there in the book. Synapses. What is the meaning of synapses? Coming together of the chromosomes. Did you see that pair of chromosome there? This one. Synapses, and now followed by synapses, chromosomes coming together, synapses, and now let us talk about this. This points you have to study. Significance of meiosis. ये टिक करो. Significance of meiosis. Textbook में that tick that part. I'm teaching you only that part. देखा? Did you get that? Meiosis part in that. Significance of meiosis. Significance of meiosis. So I'm talking about maintenance of the chromosome. This part. This part is very important for you to understand. इसमें questions आता है. So I said for meiosis, two differences you now know. Which type of cells show mitosis, which type of cells show meiosis. What about the chromosome number, mitosis, meiosis? You are clear about that? How many daughter cells, mitosis, meiosis, you are clear about that? Fourth, I am talking about if it is a cell showing meiosis, the homologous chromosomes come together. Homologous chromosome. What do you understand by homologous chromosome? A pair of chromosome where there is a maternal chromosome and there is a paternal chromosome. Uske baad, this concept of pairing is the same like mitosis that you have studied. Pairing of chromosome, duplication of chromosome. We have studied, no, in mitosis. I've given you a question also just now. Pairing, duplication happens in prophys. Let's study that. So this is the same as duplication. This is the same as duplication. But what is the difference? If I have to parallelly draw a mitosis, try to understand. This is meiosis ka stage. I am drawing a stage of mitosis. How will I draw? In a stage of mitosis, this is one chromosome and this is, in a stage of mitosis, if I have to show duplication of chromosome, I should show this way. Yes or no? But in this case, what is the difference? This is a complete pair. Because this was one chromosome from the mother, this was one chromosome from the father. Each of them got duplicated, but they are very closely placed. They are very closely coming together and placed. Unlike mitosis, if I have to draw one more pair of chromosome for mitosis, no, this may be here somewhere. Are they very closely aligned and placed side by side? No. But for a cell undergoing meiosis, this is happening and this should happen. What is this process called? Synapsis. This is followed by a process like this that you have in your book there. Please stick that diagram, that diagram comes in your paper. This process, what is this process called? Where, now try to understand these terms. So what am I showing you here? I showed you right from this stage. This gave rise to a new copy, no? A duplicated copy. This chromosome gave rise to a duplicated copy. So what are we going to term these duplicated copies as? These are called sister chromatids. These are called sister chromatids. So this is sister chromatid of this chromosome and this is sister chromatid of this chromosome. Am I clear? What am I saying? These two new copies that are formed. You may find it confusing. Let me write one. This blue new copy that I am trying to show you is a duplicated copy of this chromosome. So this chromosome will be termed as sister chromatid of this. This will be termed as sister chromatid of this one. But this chromosome, these two are non-sister chromatids. What is the difference between sister and non-sister? Try to understand. This is another sister chromatid I am saying. Now, let me number this chromosome to make it very easy. 1, 2, 3, 4. मैंने ऐसे number कर दिया. अभी now 2 is sister chromatid of 1. Please try to follow the last girl there. 3 is sister chromatid of 4. What about 2 and 3? Can they also be termed as sister chromatid? No, they are non-sister chromatid. इनका chemical composition same नहीं रहेगा ना? Gene sequence will also be different. Am I clear with this concept? This is one chromosome. This is another chromosome. When they duplicate, sister chromatid of this one. when this duplicate sister chromatid of this one. Clear? Now this is a homologous pair. So what about this two? Non-sister chromatids. These two non-sister chromatids, which are actually the duplicated chromosomes, always have a tendency of overlapping. Always have a tendency of overlapping. Overlapping is always between non-sister chromatids. They are non-sister chromatids. Confusing है कि clear है? Clear है? Crossing over. What is this process called? Crossing over. What is it called? Crossing over. What is the meaning of crossing over? Exchange of chromosomal segments. Non-sister chromatids, they always overlap each other. And then exchange chromosomes. Are you able to find the difference? What has happened here? From the blue chromosome, from the maternal chromosome, a portion of that has gone to the paternal chromosome. And from the mother's chromosome से father's chromosome में एक part exchange हो गया है. So from the maternal, a part has gone to the paternal, and from the paternal, a part has gone to the maternal. This is what is the reason for variation. This is what is the reason for variation, and in the long run, evolution, if you speak. So ठीक है, evolution we are not talking right now. So variation, what is it? In genetics again, this entire story you are going to learn. Because of this process of crossing over, that reproductive cells show we are not identical to our parents. Are you identical to your mother? Are you identical to your father? No. And do you show some new characteristics that both of them don't have? Yes, if you analyze, many. There will be at least 5 to 7 characteristics in you which neither of your parent has. How is it possible? Because of this process known as crossing over, which results in new characteristics and which is known as recombination. Which is known as recombination. So, itna hi enough hai tumhare liye to understand that the basic concept that you should know. Meiosis, one is synapsis hota hai. After that, crossing over is always happening between the duplicated chromosomes. तो वो जो न्यूली फॉर्म्ड क्रोमोसोम्स है, डुप्लीकेटेड क्रोमोसोम्स है, उसको क्या बोलते हैं? नॉन-सिस्टर क्रोमैटिड्स. क्रॉसिंग ओवर ऑलवेज टेक्स प्लेस. टिक द करेक्ट आंसर. क्रॉसिंग ओवर टेक्स प्लेस बिटवीन सिस्टर क्रोमैटिड्स की नॉन-सिस्टर क्रोमैटिड्स. नॉन-सिस्टर क्रोमैटिड्स में सिस्टर क्रोमैटिड्स में कभी भी नहीं. व्हाट इज द मीनिंग ऑफ सिस्टर क्रोमैटिड्स? इन दोनों के अंदर क्रॉसिंग ओवर हो सकता है? नो, बिकॉज़ दे आर ऑफ द सेम पेरेंट. एक मैटरनल कॉपी और एक पैटर्नल कॉपी के बीच में ही क्रॉसिंग ओवर होता है. एंड दैट्स द रीजन ऑफ एक्सचेंज ऑफ क्रोमोसोमल सेगमेंट्स. एंड दिस इज द रीजन फॉर फॉर्मेशन ऑफ न्यू कॉम्बिनेशन ऑफ जीन्स इन आवर क्रोमोसोम्स, जो हमारे दोनों पेरेंट्स के क्रोमोसोम में नहीं रहता. एंड दैट इज द रीजन फॉर न्यू कैरेक्टरिस्टिक्स दैट वी शो एज कंपेयर्ड टू आवर पेरेंट्स, व्हिच इज नोन एज वेरिएशन. अच्छा, एक और सिंपल सी कॉन्सेप्ट जस्ट गो थ्रू. व्हाट इज डिफरेंट इन meiosis? स्टेज डायग्राम्स नहीं है फॉर योर सिलेबस, रिमेंबर, बट ओनली टू अंडरस्टैंड. सो एट द एंड ऑफ meiosis, what is happening? When it is time for anaphase, are we seeing single chromosomes, or are we getting to see duplicated chromosomes? Duplicated chromosomes, no? This is one difference in meiosis. Meiosis में in the first set, इसको हम बोलेंगे anaphase 1. This is first stage. In anaphase 1, when the process of chromosome separation happens, it is just the paired chromosome going. एक full pair इधर आ जाएगा and this pair will go to the other side. So both these two pairs वो separate. अगर mitosis होता तो क्या होता? Centromere divide होता ना. यहाँ पे in the first phase of anaphase centromere divide नहीं होता है. In the first phase of anaphase, there is no centromeric division. But so far, that's why I gave you this question. The phase in which centromere divides. अगर mitosis है तो always we write anaphase, no? But in meiosis, anaphase 1 में centromeric division है ही नहीं. It's only splitting up of this. So when these 4 chromosomes come together, what do we term this as? Tetrad. ये नाम भी याद रखो. Tetrad. When all the four chromosomes are together, very close, and undergo the process of crossing over, what is this word called? This word is called tetrad. It's there in the book. When you read, you will come across tetrad. ठीक है? ये सारे words याद रखना है. And then after that, when the two new cells, so this is telophase 1. Telophase 1 में how many cells are we getting? 2 cells. Number of chromosomes वही same रहेगा. So when we talk about, see there, 2 chromosomes parent cell, and here also I am talking about 2, but it is duplicated. But we will not talk about this as 4. 2 only. Duplicated 2, duplicated 2. And what will be the fate of this cell? Again divide. Now यहाँ से जो division होगा, that will be same like mitosis. So if you have understood, now you tell me. Let me see. If you have understood, then all of you should be able to tell me. Now if I draw the anaphase stage, how should I draw the chromosome? ये वाला cell undergoing mitosis, because meiosis 2 is same like mitosis. Just try to understand and tell me this cell undergoing a similar division like mitosis, then anaphase का diagram how do I draw? Chromosomes में कैसे draw करूँ for this?
The chromosome will split apart.
Chromosomes will split. मतलब
On the chromosome.
one chromosome this side and the other chromosome this side, and one is here. Clear? आया समझ में? Yes or no? And the next, if I draw in between stages, I am not drawing. What will be the fate of this? एक chromosome इधर, एक chromosome इधर, and clear? So अभी chromosome number what should I write? 1, 1, 1, 1. No? Is the chromosome number getting half? यहाँ पे 2 था, and now I am writing 1, 1, 1. Reduced to half. Aya, roughly, did you understand what the process is? There are detailed stages. All the diagrams we can draw detailed stages है, but this is what the differences that you should try to understand. है कोई doubt इसमें? देखो, सबसे ज़्यादा ये concept तुमको याद रखना है. Crossing over, tetrad, recombination, variation. ये वाली words आया समझ में? Did you understand all these words? Any confusion? Synapses, sister, non-sister chromatids, ये तो हो गया simple concept, and then how exactly the difference is. So that whenever you are asked to write down differences between meiosis, mitosis, you are able to write. So now if I ask you, tell me 5 differences between mitosis, meiosis. बताओ फटाफट. 5 differences between mitosis, meiosis. Number 1. बोलो. First difference between mitosis, meiosis. Whatever you remember, बोलो. Whatever you remember. First difference between mitosis, meiosis. The type of cells they are present. वो याद है? The type of cells जहाँ पे process होता है. The type of cells which show the process of mitosis. The type of cells which show meiosis. अच्छा बोलो.
Somatic.
Mitosis in somatic, meiosis in
Reproductive.
reproductive. Okay. एक और difference बोलो.
Chromosomes in mitosis remains the same, and in meiosis there is half.
Okay. Third difference बोलो.
Meiosis is longer than mitosis.
The process of meiosis is longer, completed in 2 phases. Meiosis 1, meiosis 2. Mitosis is shorter. Fourth,
Meiosis are reduced in mitosis, and in meiosis polaris.
very good. Fifth,
Mitosis occurs throughout life. Mitosis.
अच्छा, throughout life का function significance बताओ. Mitosis required for growth and repair. Meiosis required for formation of gametes. एक और.
In mitosis there are full set of chromosomes, and in meiosis there are half set of chromosomes.
अच्छा, haploid, diploid. और कुछ है difference mitosis, meiosis?
It is completed in mitosis.
हाँ. एक और चीज याद रखो. In mitosis There is no variation. Cells are exactly identical. Meiosis, I just now told you, no? After meiosis, cells have recombination. Organisms will show variation. A cell which is undergoing the process of mitosis, do the chromosomes undergo crossing over variation? कुछ होता है? No, the chromosomes remain exactly the same. But a cell which is undergoing the process of meiosis, chromosomes don't have the same setup. Chromosomes have undergone so much of change, combination हो चुका है, crossing over हो चुका है, and the gene sequence is different. ठीक है? Anything more? I think that's all that we can discuss about this chapter. Quite a number of concepts to study. Few questions already I had asked you to solve. The rest of the questions I would ask you to solve from the text. All the questions from the text. Quite a number of them we have done in the class. Some of them I had asked you to do as homework. जो भी बचा है, all of you. Your book is different, no? Both of you. Book is different. Go through the questions. Go through the questions. Any question that is left out, are you able to solve all of them? Did you solve all of them? Any question left out? Any question doubtful? No? Now, if that is over, I will give you time to read. I don't know before your when is the bio test on this chapter?
Sunday.
This coming Sunday. Achha, this coming Sunday, before this coming Sunday, I don't think we are meeting. So in that case, you go through properly, read and read this part of the chapter and try to do the best. Practice some diagrams also. Absorption of roots. Let us just start this chapter as it is. Will not be able to cover much, but anyways, you can start. Let us get introduced with this chapter. Absorption by roots. What am I showing you? I am showing you the structure of the root hairs, the root structure. Now here, if I try telling you that what are some of the specialized features of the root or functions of the root. What are the functions of the root that we understand? Simply to start with, the major function of the root. What it is?
Absorption of water.
Absorption of water, holding the plant. No? So we are majorly trying to understand the concept of absorption of water. Absorption of water. And with reference to the root, straight away we come across the concept of root hair. We talk about absorption. Just the introduction we take up. We are talking about the roots. The structure of the root, whenever we see the structure of the root, every single root has got its outgrowth spread out. And these outgrowths, if you see a small region is magnified, they more or less appear like this. They more or less appear like this. What are these called? The root hair cell. The root hair cell. So now, whenever I'm talking about a simple component, what is this? I talk about the root hair cells. What are some of their special properties?
To absorb water and minerals.
No, that's the function. I first of all try to understand the structural property. The root hair cell, they will very much have the cell wall and the cell membrane. Should they have the cell wall and the cell membrane both? Should they have the cell wall and the cell membrane both? Yes, because it's a plant cell we are talking about. No, the cell wall and the cell membrane both. And the features of the cell wall and the cell membrane already you know from the previous year's concept. Cell membrane, what is the nature?
Selectively permeable.
Selectively permeable, semi-permeable. What is the meaning? Some few molecules can pass through. Cell wall on the other hand is
Freely permeable.
freely permeable. All type of molecules can pass through. So these are the basic nature. Composition wise, what is the nature? What is the difference? Cell wall, cell membrane. Composition wise, cell wall.
Cell wall is made of cellulose and cell membrane is made of lipids.
Lipid. So that lipoprotein. So that also talks about its nature. If I have to talk about the cell membrane, I will say that it is more or less extensible. Cell wall is rigid because it's made of that sugar, that polysaccharide, no? Apart from that, when we look into the structure of a cell, root hair cell, now what are the details? Structure of the root hair cell, if seen in detail, what are some of the parts? Let us try to understand the basic structure. See there, a particular structure of the root hair cell. What are the parts? It should have a cell wall, cell membrane, adequate amount of cytoplasm. Now we talk about the presence of the vacuole. Vacuole, a prominent organelle present in the plant cells, already established. No? You're acquainted to this now. 1 month already over, no? Is bell ke sath abhi tak he did not get friendly. This is such an eco-friendly bell this is. Now, vacuole. What is the membrane of the vacuole called? Tonoplast. What does the vacuole contain inside? What is that liquid inside the vacuole called? Cell sap. Why not water? Why not water? Because it's majorly water. Why not water?
Because it has some components like anthocyanins.
Because it has got minerals and ions dissolved in it. What do you understand by cell sap? Water, which has got adequate amount of minerals and ions dissolved in it. Okay. So what are the parts of the root hair cell? Cell wall, cell membrane, cytoplasm, vacuole, and a nucleus. This diagram you get in your paper and label the parts. Label the parts. So all these you'll have to remember. We'll have to stop here and we'll continue with this chapter the next day.
चार मार्क्स का लेकर मेरे आते हैं नहीं तेरे फ्यूचर चावल सारे का होता है मैंने बिना पूरे चावल करेक्ट करके चावल बोल दिया